very much. I want to up front acknowledge Dr. Carmelo Carmona-Rivera, so I don't forget to do it at the end. He is the staff scientist at the NIH and basically supervised and or conducted almost all the bench experiments that I'm going to show you today. So this is histology of bowel, affected bowel from a young child with ADA2. And what you can see in the different panels is this is just different sections of small bowel showing vasculitic involvement of medium-sized arteries at various stages. In the top left, you see panel A. That is an early stage lesion where the actual lumen is relatively preserved and where you see a clustering of inflammatory infiltrate right up here in the top right corner of this. This is neutrophilic. And as you progress, you can see various stages of damage to different arteries affected by this disease, and ending here with complete obliteration of a medium-sized artery. And so if you think about the talk that I'm about to give you at the highest level, what I'm going to tell you is essentially what happens with neutrophils potentially in this disease, and what happens as it progresses and interacts with macrophages. As you just heard, macrophages are important in the disease, and a lot of the early stage mechanistic work has focused on macrophages. And so this talk, we're really going to take a step back and look at what we think may be an earlier player in earlier inflammatory lesions in this disease and look at the role of neutrophils. So why are neutrophils important? I took this slide from Carmelo. The reason I did is because as you see the bullet points that come out, they're little neutrophil bullet points, which I thought was amazing, if they show up. So neutrophils are the most abundant white blood cells in circulation. They're terminally differentiated cells endowed with crucial roles against host defense against microbes. They're recruited from circulation into tissue in response to various chemokines and cytokines. And stimulated neutrophils can undergo an unusual form of cell death called netosis, which I'm going to talk to you about in a little bit more detail. But suffice it to say that neutrophil extracellular traps, or nets, are a meshwork of DNA decorated with antimicrobial proteins. And aberrant or enhanced formation of nets has been shown to play an increasingly important role in the pathophysiology of several systemic inflammatory diseases. And the main ones where it's been looked at is in lupus and ANCA-associated vasculitis. And these are two diseases that have a really strong phenotype that can affect small to medium-sized arteries. And armed with that knowledge, we thought, are nets playing a role in ADA2? So just to get everybody up to speed, neutrophil extracellular traps are formed when either various stimuli, sterile or non-sterile, it's the left-right, you know. So sterile or non-sterile stimuli, microbes or other inflammatory cytokines can trigger this coordinated cell death process that's different than necrosis, different than apoptosis, where essentially what happens is you get dissolution of the nuclear membrane, you get citrullination of histones, so the DNA unwinds in the nuclear core, and you get intermixing and binding of neutrophil granular proteins to DNA. And at the last step, the outer cell membrane lyses, and these neutrophils eject these chromatin-like spider webs that occupy a lot more space than the actual size of the cell. And the putative role of this process is to engulf bacteria and to facilitate their elimination. However, when this process becomes aberrant or enhanced, either by increased net formation or delayed net clearance, it's been associated with a lot of different autoimmune type diseases. So this is a picture that Carmelo took of immunofluorescence microphotograph showing nets. And so I'm going to show a lot of these types of pictures, so I just want to make sure everyone's oriented. Blue is a DAPI DNA stain, so these are neutrophils that are intact that haven't netted. And then red is staining for myeloperoxidase, a neutrophil granular protein, and so where they co-localize, you see these long, wispy pink strands. These are nets. So the first thing that we asked are nets present in patients with ADA2? And we went to pathology tissue that we had, again from small bowel, from a child with vasculitic involvement. And what you're seeing here are stains for DNA and for citrulinated histone 4. So this is one of our preferred markers in tissue to demonstrate netosis. So everywhere you see pink is nets. And you can see, and on high power, this tissue is just chock full of nets. So that convinced us that, yeah, this process was indeed happening in vivo in the tissue, but what about in circulation? There's various ways to quantify or assess NETosis in circulation, but the way that our lab prefers to do it is to measure a subset of neutrophils. These are a pathogenic subset of neutrophils known as low-density granulocytes because you can isolate them and measure them and quantify them in circulation. And so what low-density granulocytes are is when you separate blood over phycol, usually normal dense neutrophils, they're heavier, they fall to the bottom of the tube. And up here you have your PBMC layer. So this really is patting your head and rubbing your belly. So this is where you see monocytes, lymphocytes. But what a lot of groups have noticed is that this layer actually has granulocytes or neutrophils, lighter, less dense, low-density neutrophils that are not falling to the bottom of the tube. They're staying here in this interface layer. And the Kaplan lab, which our group is a part of, has characterized the function of these cells in lupus and shown that they are pro-inflammatory, that they are cytotoxic to endothelium. But the hallmark characteristic of these cells is they undergo robust spontaneous NETosis in vitro. And so here's a picture of low-density granulocytes from a patient with lupus undergoing profound NETosis. Normally, if you isolate normal dense neutrophils, you have to stimulate them in vitro with some sort of stimulus to get them to produce nets. These cells do it professionally. They don't need any stimulation. And so we started to measure and try to isolate and quantify the amount of low-density granulocytes in circulation in ADA2 patients. And for reference, we don't find these types of cells in healthy controls. And so you can see, we see them robustly in lupus, and then these were from several patients with ADA2 where we sampled blood during active disease, and this is in after treatment with TNF inhibitors. So you can see that these net-making pro-inflammatory subset of neutrophils are quite abundant in this disease and actually track with disease activity. Now, this is our working assumption that we were thinking about when we started this project a couple years ago, and I think we've heard several lines of evidence today to suggest that maybe it's incorrect, or maybe it's more nuanced than we thought, and we're learning more and more about what the role of ADA2 could potentially be. But when we first started thinking about this, we said ADA2 is a secreted protein, so perhaps it is regulating extracellular adenosine levels, and its job is to degrade adenosine or de-adenosine to inosine in the extracellular space. And so if you're deficient in ADA2, perhaps there's increased levels of extracellular adenosine. And so what we were thinking about is, okay, what is adenosine doing to neutrophils? Is it inducing NETosis? As I mentioned at the outset, there's a number of things that can trigger a neutrophil to net. It was previously unknown when we started this project of whether adenosine can trigger NETosis. And so these are neutrophils from healthy controls without stimulation, and these are neutrophils with stimulation by adenosine, and you can see pretty profound, robust NETosis. We see this across a range of concentrations, and as Dr. Hirschfeld mentioned, this is probably 0.2 micromolars is about the level of concentration you may expect to see in inflammatory sites, all the way up to supraphysiologic levels, 16 micromolars. But you can see we see robust NETosis across a range of these concentrations. So then we asked, okay, adenosine is stimulating nets. What's the role of ADA2? Can it block NETosis? And so here we're seeing no stimulus, healthy control neutrophils, stimulated with adenosine to produce nets. And here is this in the presence of recombinant ADA2, where you don't see NETosis. So put in bar graphs, this is at 0.2 and 16 micromolars. You can see profound NETosis of adenosine on neutrophils that's blocked in the presence of recombinant ADA2. So then we wondered, well, ADA2 is secreted from macrophage. Is this regulating and mediating this response? This gets to this idea, are macrophages and neutrophils cross-talking with each other? Neutrophils get turned on by extracellular adenosine. They become pro-inflammatories. Macrophages come in, secrete ADA2, and shut the process off. So to do that, I'll show you, here's control neutrophils stimulated with adenosine. And then we actually derived macrophages from healthy controls and from patients with ADA2 and took the supernate from them. So in the supernatant, you would assume that there's ADA2 in the controls, but not in the ADA2 macrophages. And again, when we add supernatant to these stimulus experiments, With healthy controls, it shuts off adenosine-mediated netosis. But macrophage supernatant from DATA2 patients, we still get robust netosis. We've talked a little bit at the meeting about different adenosine subtype receptors. There are 4, A1, A2A, A2B, and A3. And these are, in neutrophils, these are known to, A1 and A3 are thought to be somewhat pro-inflammatory on neutrophils, promoting chemotaxis, degranulation. They also increase levels of intracellular calcium, which is a key step in the netosis process. So we sort of a priori hypothesized that if adenosine is mediating netosis through one of these adenosine receptors, it would probably be A1 or A3. And indeed, it was both of them, actually. We don't have good antagonists for A2B, so we can't study that. So we only looked at A1, A2A, and A3. And here you can see that in the presence of an A1 or an A3 receptor antagonist, we almost completely abrogate adenosine-mediated netosis. With an A2A antagonist, you still get adenosine-mediated nets. And we then wanted to look at what's the receptor density look like of A1 and A3 receptors on neutrophils. So here you can see stains for A1 where it's quite abundant on the surface of neutrophils. Here's A3, less abundant but present. And we did flow cytometry, and we actually noticed kind of 3 different populations in the A1 receptors and 2 in the A3. So sort of a low, medium, and high, and a low and a high. And what we've noticed in the lab is when we took neutrophils from DATA2 patients, if they were female patients, they responded much more potently to adenosine. Adenosine induced nets a lot more potently in the female patients. So we actually started to look and see was there sexual dimorphism or differences in the amount of these specific adenosine receptors by gender. And so this is from the DATA2 patients, and you can see that female DATA2 patients had much more A1 and A3 receptors. So we extended that and said, well, does this apply? Is this specific to DATA2 or not? And so we looked at the same thing in healthy controls and saw the same pattern, actually. Female from healthy control are enriched with A1 and A3 receptors. This may have some implications in diseases that are sex-associated and associated with neutrophil net formation, things like lupus. So we think those would be quite interesting to explore. We also think it would be interesting as the cohort sizes increase in this disease to see if there is actually different clinical phenotypic expression by gender in DATA2. It's alluded to, Dr. Lee gave a really nice presentation talking about do the carriers have phenotype. I'm not going to spend too much time here other than to say that we actually studied neutrophils in the parents of these children with DATA2, and we actually did see dysregulated neutrophil function in the parents who are carriers of mutations, and we were able to actually quantify a large amount of these low-density granulocytes in the carriers. So up to this point, and then on the last section of the talk, we see that enhanced netosis is a feature of DATA2, that adenosine mediates net formation, that it's abrogated in the presence of recombinant ADA2, that adenosine receptors A1 and A3 trigger nets, that neutrophils from female patients with DATA2 and female healthy controls are enriched with A1 and A3 receptors, and that monoallelic and biallelic mutations in ADA2 are associated with neutrophil dysregulation. So in the last bit, I want to talk to you about how nets may interact with macrophages in DATA2. And so to do this, we actually looked for activation of NF-kappa B, which is a transcription factor, a master transcription factor that's ubiquitously present and when activated can produce inflammatory responses. So here, looking at the P65 subunit, you can see healthy controls. These are macrophages. When you add LPS, you activate NF-kappa B, shown in pink, and when you take nets from patients with DATA2, you get the same response. What we actually looked at next was what happens to supernatant production from these macrophages. So if you take macrophages and expose them to nets from DATA2 patients, they robustly produce TNF. And we went back to the tissue to see if this made sense spatially. And you can see here macrophages stained in green and the nets in pink. So they're in spatial proximity. These macrophages could, in theory, be ingesting these nets and stimulating this process and producing TNF. As I mentioned, I'm going to run out of time, but we think that maybe there's some strategies to decrease net formation in DATA2, either blocking A1, A3, or actually agonizing A2A, which is data I didn't show you. And we were able to actually do this. We modulated macrophage response to NETs by agonizing A2A and also with anakinra, which we could talk about later. So this is the summary figure. Stimulation of neutrophils A1A receptors and A3 by adenosine produces NETs. LDGs in this disease spontaneously produce NETs. These are ingested by macrophages, activate NF-kappa B with resultant production of TNF, and it feedback loops because TNF is a known stimulator of NETosis in and of itself. And so that's how we've been putting things together. Thanks.